Hi everyone, thanks so much for joining. I think we'll get started. We've been seeing a few more people popping into the lobby, so I think we'll just keep adding those as they come in. My name is Desley, I'm the head of sales here at AGRF, and I'd like to welcome you to today's Short and Seek webinar in our Short and Seek series. And today we're talking about polygenic risk score. I'm going to just give you a brief introduction to AGRF, and then I'll hand to our expert, Melinda Zeno, who will go through the actual PRS that we're offering. So again, welcome. I'd just like to start by acknowledging the Wurundjeri Woiwurrung and Bunurong Boon Wurrung peoples of the Kulin Nation as the traditional owners of this land, and pay respect to their elders past, present and emerging. And we especially pay tribute to their profound understanding of the natural world, which encompasses a deep knowledge of the life sciences acquired through careful observation, experimentation and storytelling, passed down through the generations over tens of thousands of years. So just a little bit about AGRF. We're Australia's largest genomics service provider with more than 25 years of delivering genomics excellence. We were formed as a collaboration between the Walter and Eliza Hall Institute and the University of Queensland more than 25 years ago, and we're an independent not-for-profit company. It was set up as a collaborative unit that could apply for major grants and bring in technological advances to Australia that people may not be able to afford on their own. We have a national network providing Australian scientists, researchers and clinicians with access to these robust and advanced cutting-edge technologies. And we are ISO 15189 accredited for clinical services here in Melbourne, and also 17025 for all of our other services across the country. And that's an accreditation for biological testing. So we're covered for human pathology, animal health, and the life sciences. We do have a strong national collaborative network with researchers and diagnostic organizations across the country, and we have a track record of adopting technologies early on. Things like the Promethean from Oxford Nanopore, the PacBio Revio, and the Illumina NovaSeq platforms as they've come out. To show our national network, we are hosted by these institutes in Adelaide, Brisbane, Melbourne, Sydney and Perth. With more than 100 highly skilled staff across these 5 sites, we are working with more than 450 research and commercial organisations each year. And we have a dedicated innovation and development team. That team's responsible for collaborating with our clients to help bring new technologies into service. We also have a dedicated bioinformatics team, and we serve more than 3,000 clients and collaborators each year. Just wanted to give you a very broad overview of our comprehensive suite of services. So we're able to provide end-to-end services for a lot of applications, from nucleic acid extraction all the way through to delivery of data from our bioinformatics team. We have Sanger sequencing in every state across the country, all of the host institutes mentioned, I should say, as well as we also have next generation sequencing, long read, arrays of genotyping, which Melinda is going to talk about today, as well as protein biomarker discovery that we've just added with the Olink technology. I also want to briefly touch on our 15189 accreditation for clinical genomics. I've listed the services here. I won't go through them all one by one. You can read through those yourself, but I would like to just mention that we have a quality and regulatory affairs manager who makes sure that all of our accreditations is up to date. She also manages our quality management system and all of our quality assurance programs that we go through. So everything is done to a very highly stringent capability. So again, here showing the clinical services that we run. These are all run in Melbourne, from extraction through to whole genome and whole exome sequencing, our global screening arrays that Melinda's going to talk about today, some cytogenetics array, as well as variant confirmation through Sanger, and our newly added TrueSight Oncology 500 for oncology applications. So why did we add polygenic risk scores to our suite of offerings? We have been working with a lot of clients who are developing their own polygenic risk scores, and those are fantastic. Those are set up for applications that aren't necessarily available in any other off-the-shelf system. So working with clients with things like glaucoma, melanoma, and also some risk scores within the wellness industry. So we've definitely seen an evolving market demand for these predictive personalized insights. As people become more knowledgeable about genomics, they're hearing the words more freely in the media, people are talking about it on the news, and the government is starting to rebate more and more genomic tests, which is fantastic to see. So that's one of the reasons. We also saw the value in unlocking existing genomic data. So being able to go back and take your array data that you already have, any SNP or genome sequencing data that you already have, and being able to run that through a polygenic risk score pipeline and understand the risk across a population. I think that's really valuable and one of the reasons why we've added it to AGRF services. There's also expanding clinical wellness applications. We're seeing a lot of interest in more genomics for people who just would like to know more about their health and what they can manage themselves. And we see that happening through direct-to-consumer applications. And finally, I think this is my favorite, but the opportunity for ethical and inclusive innovation. So what I mean here is a lot of the databases that we use for our data when we're doing biobank mining doesn't necessarily include the hugely diverse and population that we have here in Australia. So to be able to start using Australian data to build up that information, to be able to potentially be added to some of these arrays in the future to represent more ethnicities across the world, I think that's a really valuable exercise. So I think that's it from me. I'm going to hand over to Melinda. Melinda is our genotyping manager. She's been at AGRF, I don't think she'll mind me saying, she's been here for 25 years and is definitely our expert in genotyping. So Melinda's going to take you through the genotyping service as well as the polygenic risk score service that has now been added to that. So over to you, Mel.
Thank you, Desley, for that great introduction. So today I'm going to be talking on polygenic risk score. So today we'll cover what is a PRS, how is a PRS calculated, the applications that you can see and use these new testing in, and a little more insight into AGRF's new PRS service that we are offering. So for those who don't know, a polygenic risk score, or PRS, estimates the genetic predisposition to complex diseases by analysing multiple SNPs. It works by aggregating the effects of the thousands to millions of variants within our genome. It's derived from either that whole the GWAS data that you might already have, or the sequencing data, and it analyses the risks by effect. So what it can do is support that disease prediction by risk stratification. And why does it matter? Well, PRS can enhance the precision medicine strategies that we currently have. It is an extremely cost-effective and standardised genetic risk assessment, and it can really integrate seamlessly with the genetic data people already have. And the report is quite a simple report because it reports the percentile risk that does predict that genetic predisposition to that complex disease. So why would you use a PRS? For once, it is a one-off test for the lifelong genetic risk of that disease. So what it can be used is early impact and early risk detection. So it can be used in either the research setting, the clinical risk assessment, or what we touched on earlier, that personalised medicine. So in terms of disease risk, it can look at cancer, cardiovascular disease, diabetes, and really a multitude of complex diseases that are in our community. As I mentioned, it can enhance that screening, but also that early intervention. So some of these diseases like diabetes, early intervention might mean that environmental or lifestyle changes for an individual can lower people's risk of developing these diseases. And with that, you can integrate this genetic knowledge with other clinical risk factors. So the impact of having a PRS is that early risk detection, the personalised prevention, but also better clinical decisions, because it can improve those risk models with the genetic insights that we would now have. People can get those targeted therapies, but also on that population level, it can enable the resource allocation and proactive care at a more global level for communities. So in terms of the key features of AGRF's PRS service, look, there are a lot of inbuilt and in-house models that people have developed based on their own large-scale studies, but the AGRF's new offering, which is partnered with the Allelica Predict pipeline, is a validated and standardized PRS model that is suitable for diverse ancestries, which is often a limitation of PRS models. It's customizable. It's also a cloud-based platform, and it really does give those clinical insights to integrate the genetic risk with actionable data. So in terms of the disease prediction, the AGRF offering covers 20 common complex diseases, from oncology to cardiovascular diseases, metabolic and endocrine, and other complex diseases such as Alzheimer's and osteoporosis. I'm not going to go through all 20 diseases on the list, though if your disease of interest isn't on the list, please do contact us, because the Allelica pipeline, we can look at exploring customized PRS solutions based on published GWAS data, or also your own data that you might have for GWAS studies. We can look at developing a custom PRS solution for you. So in terms of the data inputs that we do support, AGRF services can take from your raw sample, so from DNA extraction right the way through to PRS calculation. We can offer the full workflow from sample to the PRS. Though if you already have data available, this great service can use the array data, so you can use the Illumina global screening data, or even whole genome sequencing data. And the PRS can use whole genome sequencing data with a minimum of 2 times coverage for accurate variant calling. In terms of existing data, we can look at the Illumina IDAP files, but we also support the Thermo Fisher Axiom arrays. And as I mentioned, whole genome sequencing data. So all you need to do is submit that data along with your meta data. So in terms of using this data, as we said, we upload the raw data. So in terms of arrays, it would be the IDAT or the .cell file, or the raw FASTQs from whole genome sequencing. Now, the next point is actually quite an important one. We would next run it with the appropriate PRS model that you would like processed. But more than one of those models can be selected at no additional cost. So if you did want all 20 complex diseases from the list, you can certainly choose all 20, and it would be the same cost for all 20. AGRF run the quality control and imputation on that data, and the PRS computation, and gives a risk percentile assignment. So what that means is individuals are stratified into 2 categories, either not elevated or high risk, based to the general population for that disease. So in terms of that, as I said, your comparison is to the general population, and it does indicate the relative risk. That the characterization is into two thresholds based on that specific disease. And as I mentioned earlier, the percentage is a lifetime risk of the disease, and it's the fold change relative to the average population. So in terms of the output AGRF provides, we provide a batch CSV report summarising these PRS outcomes for all samples within an analysis batch of samples provided. So just a general overview of the workflow. As I said, From the genetic data provided, we would provide the imputation, the PRS computation, and the report generation. Though it is really important to note that PRS is not a definitive answer for developing a disease, and it should be integrated with other risk factors such as lifestyle or environmental factors. So just in summary, PRS has a real power to unlocking genomic data. It can empower personalised medicine to tailor those risks for better health outcomes. It is important for informed decision making, and it can really support researchers and clinicians in early detection, prevention and treatment of these complex diseases. And it really does advance precision health, because integrating genetic risk does turn that genetic data into actionable strategies. Strategies, and it turns that genomic data into real impactful information. So thank you.
Thanks, Mel. That was a really great summary of our PRS service. And handily, we currently have a promotion running. So we're really keen for people to see how this service can really help unlock your data insights. So we're offering the service at a reduced price for you to send in your existing SNP array data or whole genome sequencing data. And remember, that's Illumina arrays or Axiom array data, as well as whole genome. And we'll do the polygenic risk score for just $75. There's a QR code on your screen now, so you can scan that if you'd like to have a look at that promotion and all the details. I think now we're going to open up to questions. So feel free to turn your camera back on if you want to, or raise your hand and we can answer those questions for you. You can also pop your questions into the chat if you don't want to speak. That's totally fine. So we will switch out to our cameras so we can talk to you.
Hi, Desley. I have a question. It's Oliver here from AGRF. You mentioned, Mel, that the risk score can be a lifetime analysis that you can do and use to inform decisions in future. Now, as research evolves, you do get more identification of more markers over time, new markers over time that might be really impactful as well. So what happens in that case? Does it get reanalyzed, or is that something that's purely subjective? How is that talked about?
So in terms of these complex diseases, obviously there's not one SNP or one gene that's causing these complex diseases. So for most of these, it is looking at an aggregate of all known SNPs and genes that are causation for these complex diseases. So there is imputation of the data that's provided to be able to give that weighted risk compared to the population. Now, in the future, if there was a new SNP or a new gene that is known to cause these diseases, obviously you would need to re-look at this data, but this is based on, obviously, with these 20 complex diseases, years of research for these gene and SNP causing implications of these.
And Mel, usually it's millions of sample points, is that correct?
That's correct. It's not just one SNP or one gene. These are complex diseases with a multitude of genes and SNPs that all are interacting for that cause.
Thanks, Oliver. Does anyone else have any questions? Okay, if not, then we might close here. Do feel free to email us if you have any questions or contact the customer care email address on the website. But for now, I will say thanks so much for joining. We really appreciate it. And we'll see you in the next Short and Seek seminar. Thanks, everyone. Thanks, Mel.
No, thank you.